So it automatically brings to the next question as to what are the steps put in place by the FDA to weed out or identify genotoxins very early on in product development. The standard FDA approved assays are Ames test, chromosomal aberration, micronucleus or mouse lymphoma assay. Ames, as we all know, is a bacterial test involving bacteria in a test to identify genotoxins for humans is highly debatable and is a raging controversy. Bacteria have a DNA repair mechanism but is strikingly different from that of what you see in a mammalian cell. Chromosomal aberration and micronucleus tests are in fact tests which are carried out on mostly on mice or rodents, and they are not always done in a high throughput manner. So there are several limitations associated with standard genotoxic assays put in place by the FDA. And we felt that the limitations are very many, and the assays are quite time consuming. Ames, chromosomal aberration and micronucleus tests can take days and in fact sometimes weeks to carry out. The incidences of false positives are quite rampant. In fact, there are a growing number of publications which have characterized the genotoxic potential of pharmaceutical drugs which are in market right now. And if you look through those publications, you find that many of the compounds are positive in Ames but negative in chromosomal aberration or micronucleus test, or positive in micronucleus but negative in Ames. So, and these are pharmaceutical products that are available in the market right now. So there is a lot of inherent bias in the system right now, which we hope our technology could correct. The use of animals for initial screening could be possibly avoided by the technology that we have developed. And we also felt that we could add more to the science behind a particular assay by revealing the underlying mechanisms that could possibly contribute to the genotoxic stimuli or the genotoxic readout which you finally see. And the assays that are currently in practice are not always done in a high throughput manner. So there is a growing need for a human cell-based in vitro high throughput compatible genotox screen. And towards this, we decided to look into how a human cell responds to genotoxicity, and we found that there are many pathways that are triggered, that could be triggered by a genotoxin, depending on the kind of stimulus that the cell is exposed to. However, most of these signals converge at P53, P21 or GAD153, which are three genes which are central to any genotoxic pathway, which was quite interesting to us. So we decided to look into P53, P21 and GAD153 and possibly use these three proteins as sensors of genotoxicity. So what we decided to do was to pull out the promoters of these three genes or the DNA binding elements of these three genes and fuse them to a very well known protein called luciferase, which is a very well known reporter. So the Antham's genotox assay has multiple components, but in a single slide, the features are: it is simple, but it is robust, rapid, and is high throughput compatible. It could possibly reveal the mechanisms underlying the genotoxic event, because you know the genes which are involved, and by fusing a particular reporter to the promoters, you know which of these genes are activated. The cell line is a human cell line, but I should point out here that these three cassettes, or the triple sensor system that we call it, could be transferred to any cell type of your choice. So you could transfer it to a neuronal cell, you could transfer it to a gut cell, you could transfer it to a breast carcinoma cell, any cell line of your choice. It's luminescence based, so the readout is going to be simple. All you need is a simple luminometer, and the expressions are do drive the early DNA damage response elements, so you actually pick out the first signals that the cell is exposed to. So this is a snapshot of what our cell type would look like. So it's a single cell which has three sensors: P21 promoter fused to firefly luciferase, P53 DNA binding element fused to renilla luciferase, and GAD153 promoter which is fused to beta galactosidase. So you have three different kinds of reporters fused to three different kinds of sensors. So in essence, what you get is simple luminescence when the cell is hit by a genotoxin.